LabKey server has long supported being able to look up from an assay into samples to retrieve metadata and track usage of samples on the assay side. And in 16.3, we improved on that in a number of ways. We made it more flexible in terms of the locations it's able to look up into, so that not all the samples have to be in the same folder as the assay data that you are importing. And we also added capabilities to make it easier to look up across sample sets in case your assay may be consuming a variety of different types of samples. So what I've done here for this demo is I've set up a project that has a number of sample sets defined up at this level. Some of them have samples imported at the project level, while others have data imported at the subfolder level. So let's take a quick look at those so we understand what we're referencing. We have a blood draw sample set that has the primary draw information for a variety of different subjects. And then we have a aliquot sample set for the vials that are split out from those original draws. And then we also have an RNA extraction that consumes some of those aliquots, but there's no data loaded at the project level here. It's all loaded at the subfolder. So let's go ahead and start using that from the assay side to understand how we can link the samples in. So I'm going to go ahead and go into one of these subfolders where I'm actually referencing these. It's a common pool of donors, but then individual projects based on different diseases to link back to those samples. So first I'm going to show a cell count assay. So this one not only is going to reference samples that are loaded at the project level, but it's going to reference a mix of those aliquots and the original draws as part of the data. So I'm going to go ahead and go to import data. You can see I already have one run loaded. I'm going to upload a file, an Excel file from my local machine here. Grab my cell count and proceed. And if I click through to look at the data, I can see that I have a mix of the original blood draws and the aliquots, the individual vials in my sample data. So if I click on any one of these, it's linked automatically back up to the project level. I would have gotten an error if it couldn't resolve it. And I can see the history and the derivation of that aliquot. So I can go back to the parent sample, for example, and trace that lineage. I'm now going to go to my other assay, which is configured to point to the RNA sequence, the RNA extraction that I've loaded into this folder directly. Again, these are child samples of those aliquots. So if I can go to import data, different assay design here, I'm going to import QC2, finish that import. And if I click here, I can see that I have two different columns, both targeting that same RNA extraction sample set. And again, if I click on any of these, they'll take me back to the information. You can see that it is, in fact, in this SARS subfolder, not up at the project level for this derived sample. But if I go ahead and click back to the aliquot that it came from, it takes me up to the project level. And likewise, I can continue to drill back all the way to the original blood draw. So let's take a look briefly at the setup for that assay design. So if I go to my cell count assay and edit its design, you can see, so this is the assay that is looking up not to a specific assay, or excuse me, not to a specific sample set, but to samples in general. So it's configured to resolve to the EXP materials table, which is effectively a union of all sample sets. And even though the primary key of this table behind the scenes is an integer, we're importing based on the sample name and resolving that as part of the assay import process. And if I go back to my QC assay design, this one is set up a little bit differently. It is knowing that it's expecting only that RNA extraction sample data. And so these columns are configured as lookups directly to that sample set. So the new rules are that we look not only in the current folder, but also at the project level and all the way back to the shared project as well when resolving samples even if we're targeting the exp.material union table. So as you can see, we've added a lot of flexibility, made it a lot easier to organize the data based on the research needs, and still have all of the links automatically happen between the assay and the sample set.